So hello and welcome to the next in the Journal of Clinical Investigation series, Conversations with Giants in Medicine. I'm the editor-at-large for the JCI, Ushma Neal. You're in for a treat today as we get to spend the next hour with a diabetes pioneer, Dr. Dan Drucker of the Lunenfeld Tannenbaum Research Institute of Mount Sinai Hospital and the University of Toronto. Drucker's early work explored the biosynthesis, secretion, and action of glucagon, and he later went on to delineate the novel mechanisms of action of glucagon-like peptides 1 and 2. GLP-1 and 2 agonists, as well as his work on DPP-4 inhibitors, provide the foundation for some of the largest sector of drugs for both gut disorders as well as type 2 diabetes. So thank you so much for being here today, Dan.
My pleasure, thank you.
Do you think we could start at the beginning? Would you mind telling me a little bit about your parents and what you were like as a kid?
Sure. So like some of your previous guests, my parents were Holocaust survivors from Europe. They found each other in Israel after the war and moved to Montreal in 1953. And I grew up in Montreal until I was about 13 years old. And my dad then got a job for the federal government, which is based in Ottawa. So off to Ottawa we went. As a 13-year-old kid, I had no choice but to tag along, and really was in Ottawa until I left Ottawa for medical school in 1976.
So what did your parents do?
My dad was an engineer and an architect and a builder. And you may be familiar with the history of Quebec in the 1969 and 70s. There was some civil unrest and things were not exactly stable, and a lot of people were leaving, and he just found it hard to make a go of it, frankly. And so he thought a job with the federal government provided security. And my mom was trained as a dietitian. She never really finished formal school, had to leave school when she was very young because of the Second World War, but she was a nurse's aide and a dietitian, and then later on transitioned to interior design. So she basically did whatever she could to make a living.
So where did your interest in science come from?
I think that came later in high school. I just kind of noticed that when I would write an essay in English, for example, the way that it was received was highly variable. Some teachers would love it, and other teachers were not so sure I had understood, and I always remember asking myself, how did you know what Chaucer was thinking? And how did you know what Shakespeare was thinking? Why did I not convey the thoughts properly? But then science, there was only one right answer, as boring as that is, whether you do chemistry or physics or biology. These are the laws of science. And so it was much easier for me to do well. And maybe I was insecure and unable to extend myself in the humanities, but I just kind of gravitated to science.
So what was the path then via University of Ottawa to medical school?
So back in those days, one didn't have to be particularly well-rounded or highly educated to get into medical school. We had some entry-level programs, or one could apply after 2 years of college, and that's what I did, and ultimately ended up at the University of Toronto in 1976.
So what was it about internal medicine and then endocrinology that intrigued you? Where did that fascination come from?
So I think endocrinology came first. I was just flipping through the elective catalog in 1976. Younger viewers may be amazed to know there was no computers or electronic databases. You went to the medical school office and there was a big thick book of electives that you had to flip through. And endocrinology caught my eye, all these really cool diseases. And there was a brand new chair of endocrinology at the time. His name was Jerry Burrow. He had just arrived from Yale. And I did my first elective with him. And I think that was it. I mean, he had a great personality. He loved teaching. He had a lab as well as a clinical practice. He was enthusiastic about everything. There was no negativity about him whatsoever. And it was just fun to do that. And that just led to a series of endocrinology electives. And by default, if one wants to do endocrinology, one has to do internal medicine as well. So that was a secondary consideration.
So the fascination with endocrinology, from what I read in my background reading, that kind of turned into a fixation on the thyroid.
Yeah, so my mentor, Jerry Burrow, was a quotation marks thyroid person. That's what he studied in the lab. In fact, I did some experiments with him looking at phosphorylation of thyroglobulin and ovine thyroid cells in culture. And initially I thought it was really cool and didn't really understand why I was given 5 millicuries of P32 and everyone else was hiding outside the door. Learned later, and happily had 3 healthy children. But he just loved the thyroid, and his practice was rewarding people presented with very treatable illnesses, and the science was really cool. And so I think it was just Jerry Burrow and his leadership in the thyroid area that became my interest. I adopted his interest.
So then if you've got this interest in the thyroid gland and perhaps a thought about the clinical side of things, what led you to going to work with Joel Habner at the Mass General?
Yes, so that was all directed at thyroid. I interviewed at 3 great labs. So I interviewed with Bruce Weintraub at the NIH, who had cloned TSH, really exciting. And I interviewed with the legendary Seymour Reichland at Tufts University, who was an expert on thyroid hormone releasing hormone. And Joel Habner at the Mass General had a grant on glycoprotein hormone genes and TSH as well. So all of these interviews were directed at thyroid training. And, as the story goes, I accepted Joel Habner's offer because he had, I think, the most experience in molecular biology, which at the time in the early 1980s was the up and coming sort of area to be in. It had displaced monoclonal antibodies as the really cool new science. And when I got to Joel's lab, very simply, he said, well, Bill Chin is spearheading the thyroid project, and he's leaving to be independent at the Brigham, so you'll just have to work on proglucagon. And in the words of many trainees, I probably said to myself, what just happened? Like, I was supposed to work on the thyroid. That was very clear to everyone, and here I am working on glucagon. But in those days, one did what one was told. Basically, there was no concept of questioning the professor. I was in no position to really question the direction. And I really wanted to learn science and molecular biology. And the reality is it didn't really matter that much if I learned it in a thyroid system or learned it in a glucagon system. So I just set to work doing what I was told to do.
So at this phase in your career, had you started thinking about, being a physician scientist, or was this research elective just that?
No, this was a full-time 3-year fellowship, and I had hoped that I would be successful. I was pretty naive. I had never done any bench science other than what I described to you, working on, thyroid cells and tissue culture. Really, when I opened the book, we had this manual of molecular biology that some of your older viewers will remember was written by Tom Maniotis. It was a blue binder. And, I opened the book and started making solutions. And the first thing I looked at was like, what's a 1 molar solution? What's, mass over molecular weight? And I was literally that naive. I really had no clue what I was doing. And so it was pretty painful at the beginning. And it was literally 6 months later, after a lot of stumbling and fumbling and messing up, that Joel Habner, I finally had some data, and he looked at me and said, this might work out after all. You actually might accomplish something and be successful. And that was my first indication that, from his view, there was a huge possibility that I would be a failure, which never really occurred to me. I was ex-chief resident. I had interned at Hopkins. I had always done really well. So the concept that I might be a failure at something was not clear to me, but it was clearly apparent to him from my initial stumbles in the lab.
So what was the project that you ended up taking on in the Habner lab?
So there were a couple of projects. So we saw the sequence in the genes that had been cloned, and we really had no idea if these theoretical glucagon-like peptides, as they were designated, were they liberated. So my first project was simply to express the proglucagon cDNAs in a bunch of cell lines from fibroblasts to pituitary cells to islet cells, and say, do these cells have the machinery, what we now know as prohormone convertases, but did they have the molecular machinery that allowed them to liberate the glucagon-like peptides? And so obviously the fibroblasts didn't, and the pituitary and the islet cells did. And for the first time, we could see that not only was GLP-1 and GLP-2 liberated, but there were multiple molecular forms of GLP-1, which became important later. And then in parallel, my related project was to figure out what GLP-1 did. And that was very simple, just dumping GLP-1 on a whole bunch of cell lines, including the 3 that I mentioned, and then looking for bioactivity, changes in gene expression, proliferation, cyclic AMP formation. And we were just very fortunate that islet cells were among the cells we tested, and that's how we figured out that GLP-1 stimulated insulin secretion.
Okay. So during this 3 years, you start to have some success. You're getting the molecular biology techniques down. You're starting to find out what the actual biology is on GLP-1. At what point did you decide to start looking for faculty positions on your own?
So I think it was always understood that I would go back to Toronto, and my wife's family was from Toronto, and we really loved Toronto, and the University of Toronto was a great place. So that was my initial focus in terms of recruitment. I did look at one other university, and that was the University of Alberta, also, very famous for diabetes research. And at the time, they had the Alberta Heritage Fund, which was money taken from the oil revenues. Alberta is a little bit like Canada's Texas. And when the oil prices are high, the revenues really accrue to the government. And they set up, a $1 billion medical discovery fund to help fund scientists. So I thought I owed it to myself to go out there and look, and they were fabulous. And I think everything about the Edmonton offer was twice as good as Toronto in terms of the space and the salary and the startup funds. But I think I was just torn to Toronto and my wife's family, and I loved it there. So that's how I ended up back in Toronto.
So talk to us a little bit about what it was like starting your lab then. Was it easy? What were you balancing clinical work as well as lab work and building that? What was the funding climate like for you?
So I don't think it's ever easy. I think the funding climate, to pick up on that, was better then than it was now. So if a new investigator wrote a grant in the mid 1980s, the funding payoff was probably 25 to 35%, which is obviously much higher than it is now. And yes, I had to generate most of the initial data myself. So I had 1 day in clinic and 4 days in the lab, and I did 1 month of general internal medicine and 1 month of endocrinology consult. So it was pretty hectic. And obviously I had one child who was about 4 years old and another one on the way. So there was the family life, the clinical activities, the starting up the lab, which I think is very familiar to all clinician scientists today. Sort of jack of all trades, master of none, always feel inadequate at most things that we do.
I've seen you write frequently about the colleagues and the support that you were able to get, especially as you were establishing new mouse lines and rat lines and reporters and the kind of quality of colleagues and the milieu in which you found yourself.
Yeah, Toronto is a wonderful environment for science. We only have one medical school, the University of Toronto, and a city of Toronto size that's bigger than Boston that has multiple schools, that's bigger than Philadelphia that has multiple schools. One medical school is unique, and that means that everyone's appointed to the same school, and we don't have a competitive internal, set of issues. And, I had a wonderful collaborator, I still do, in the Department of Physiology, Patricia Brubaker, who was also training and working in the area of glucagon-like peptides. And I had a really amazing mentor beyond Jerry Burrow and beyond Charles Hollenberg, a mentor named Lou Semenovich. And Lou was the founder of the Hospital for Sick Children's Research Institute. He founded the Lunenfeld. He was a really amazing scientist, Royal Society member of the National Academy of Sciences. And he had a daughter Kathy Siminovich, who was also a clinician scientist who was 1 year ahead of me. And when Kathy trained at the NIH and came back, Lou saw that she didn't really get a lot of help with her grants and the molecular biology. And so Lou sort of adopted me as his scientific son, so to speak. And, he would say to me in, 1988, Drucker, you have to make transgenic mice. That's what everyone is doing now. I had never worked with mice before, didn't know how to do that. But I said, well, if Lou tells me that I have to do that, that's what we did. And then a few years later, in 1993, said, everyone's making knockout mice now. You have to make knockout mice. Knock out your favorite gene. I said, well, I guess I'll have to figure out how to do that. And these kind of, mentors and the advice you get are just priceless, because all of those experiments were fundamentally important for my career and the process of discovery.
Let us then turn our attention to GLP-1. So you had a front row seat. I mean, you were involved in much of the early biology, but in turning GLP-1 into a therapeutic target. So did you have an inkling that that could be something that was translational?
Well, I have to say I had an inkling because, my notebooks in Boston disappeared. And when I asked Joel Hebner, where are my notebooks? He basically said, well, we're filing a patent, to potentially protect the idea that GLP-1 could be used to treat diabetes. And, I was pretty naive. I didn't know very much about patents. Those were the early 1980s. So we didn't really have the biotechnology industry that we do today. The technology transfer offices at many places were either bolted on to the vice president research or really not in existence. And I just said, okay, well, that's fine. But I think Joel deserves a lot of credit. He had that vision that these discoveries that were made in his laboratory might not just be interesting biology, but might one day support the development of a new field. And so I was a bystander for that. But, there's an expression in medicine, see one, do one, teach one. And so after seeing those first GLP-1 patents, once we cloned the Exendin-4 gene, I said, bingo, this might be useful. Once we sort of figured out that DP4 inhibitors had really useful effects to lower glucose, that led to a dozen DP4 inhibitor patents. Once we identified the sequence of GLP-2, that led to, about 18 patents for the use of GLP-2. So I was really in the right place at the right time to watch Joel and see how technology transfer worked. And then once I was in a position to do that independently, it became much easier for me.
Right. So let's talk a little bit more about DPP4 inhibitors and your work in that whole realm. Can you shed just a little bit of light on how you and others came across this as a potential class of therapeutics for type 2 diabetes? And the last I looked, the market share for these kinds of inhibitors is massive.
Yeah, so this was a great story. And I think, the earliest description that DP4 cleaves GLP-1 and GIP, for example, was made by Rudolf Mentlein in Germany, and that was 1993. And then very quickly thereafter, Tim Kiefer's group with, he was a student at the time working with McIntosh and Peterson and Jens Holst and Carolyn Deacon and many others, as well as industry, particularly Novartis, jumped onto this concept. And I was a little bit late to the game, honestly, but I gave a talk at Tufts University, probably about 1990 or so, and I showed the sequence of these glucagon-like peptides and mentioned that they were degraded by DPP4. It was probably 1993 or 1994. I had read the papers from Mentlein, and I had a scientist wait for me at the end of my lecture, and his name was William Bichovchin at Tufts. And he had been working on DPP4 for years. In fact, had a company that was developing DPP4 inhibitors for the treatment of cancer. And he was fascinated by this glucose GLP-1 story. And he said to me, if I send you some of my DP4 inhibitors, can you test them in your mice and look at glucose and insulin and see whether they in fact are useful? And so I said, sure. I'm not even certain we had a technology transfer agreement in place. I think we had a technology transfer agreement in place after we got the really cool data. That's how we did stuff back in the day. But that led to a dozen patents with Bill Bichovchin to use DP4 inhibitors as a therapy for diabetes. And we weren't the only one that had patents. There were lots and lots of people. People in East Germany had patents, the Vancouver group had patents, and these were all blended into a master patent bank that almost every company licensed when they developed their own DP4 inhibitors.
I was absolutely fascinated by your first person article that you wrote about GLP-2 and teglutide and the whole, like, where it came from and how it developed and how long it was, and your fight to get industry interested in this as a target. So I would love to hear just a little bit more from the personal side around what were the ups and downs of target discovery through discovering that it, or like realizing that it could have some translational potential, to getting industry interested through, you know, the competition from Japanese colleagues and everything through to potentially having champagne once it was finally FDA approved.
Yeah, on a plane going to Italy. So it's a pretty simple story, actually. It's embarrassingly simple. So I had been schooled in gene transcription in Joel Havner's lab. In fact, the first couple of grants that I wrote were the control of glucagon gene transcription, and we worked on that in the islets quite a bit. But then it became apparent it would be really interesting to look at glucagon gene transcription in the gut. And to do that, we needed a gut cell line that expressed the proglucagon gene, and there wasn't one. So, you know, following up on Doug Hanahan's use of SV40 T antigen to immortalize cells and make islet cell lines, I just made a transgenic mouse expressing SV40 T antigen under the control of the glucagon promoter. And lo and behold, I got intestinal tumors, and we used those tumors to isolate a cell line called the GluTag cell line, glucagon T antigen cell line, to do transcription. Now, in order to make that cell line, we passaged those tumors in nude mice, and the tumors got, you know, a nice size subcutaneously so we could dissociate the cells and study them. But when we looked at the nude mice and opened them up, the intestines were huge. I'm not talking 15%, 20% bigger. I'm talking like 2 to 3-fold bigger. So you didn't need statistics, you didn't need fancy math to see this is a huge biological effect. And as a clinician scientist, if you go into the human literature, there are examples of humans with glucagon producing tumors who present with small bowel obstruction. You take out the tumor, the bowel abnormalities go away. So we figured there must be something that the tumor is making that's promoting bowel growth. And we figured it's probably connected to the glucagon gene. So we simply synthesized all of the glucagon-like peptides and the intervening peptides, from glucagon to intervening peptide 1 and 2, glycentin, oxyntomodulin, GLP-1, GLP-2, and we injected them into mice. Now, I had looked, as you intimated, for funding for this. And again, this was in the days where email was not really a thing, and you couldn't get the email addresses of people who worked at companies anyhow. So I sent off about 30 FedExes to big pharma and maybe some biotech companies like Amgen or Genentech. And I said, I have this model. I can promote bowel growth. I can probably figure out what the bowel growth factor is, and we can use that as a treatment for inflammatory bowel disease or unrepaired fistulas or short bowel syndrome. Very few of the companies responded. Most of them said, well, we'll bring this up at our quarterly committee target review thing. But there was a biotechnology company in Toronto, in the suburb Mississauga, where the airport is, and they said, you know what, why don't you come on over and give us a seminar and talk to us. I did. After the seminar, they said, okay, we'll give you $100,000 to do this. And that's how I got hooked up with Allelix. I don't even think we signed a contract for the work. It was just one of these, you know, things were more informal back in those days. We ordered up all the peptides. As I was getting ready to inject them, Allelix made me aware of a patent filing from Japan that you hinted at, and they had identified the glucagon-like or related peptide that promoted bowel growth, and it was glycentin. And so I was a little bit bummed out. I was partially relieved because there's a lot of pressure. This was my first company sponsored grant, and there's always pressure to, deliver. But I said, we have the peptides anyhow. They had just come in. I will just inject them into animals. We will publish a paper on whatever the results show. If I confirm that it's glicentin, that's fine. So I did the experiment. These were the days where I did these experiments myself. And it's true that glicentin was a bowel growth factor, but it turned out that GLP-2 was even more potent as a bowel growth factor, like much more effective. And for reasons that are not clear to me, the Japanese folks never studied GLP-2, and there was no real described biological activity of GLP-2 in the field whatsoever. So this led to a series of GLP-2 patents, and we synthesized 100 analogs of GLP-2, looked at the most robustly effective positions of GLP-2 that were important to substitute. Certainly the DP4 cleavage at position 2 was clear, and that again just led to, I think, about 18 patents for GLP-2, which Allelix sort of was happy to support. But to be honest, they sort of slow walked the GLP-2 development program because, coming back to a coincidence, their lead molecule was recombinant parathyroid hormone. And osteoporosis was a much bigger market than, obviously, short bowel syndrome. And so they were kind of developing GLP-2, but very slowly. And all of their effort and all of their finances was on PTH. But they ran into some toxicology trouble and hypercalcemia with that program. So then the GLP-2 program was back on.
I read in your first person narrative that you were on an exercise bike the first time you heard of approval, at least from the European agencies. So yeah, after, some stops and starts and, enrolling more and more patients and with short bowel syndrome, you did finally get to then also FDA approval, yes?
Yeah, so the European approval came first, which is a bit unusual because the FDA is usually known for being a bit ahead of EMA, but that approval came first. And I literally was at the American Diabetes Association meeting, I think in San Francisco, and an analyst who covered this area emailed me and said, congratulations, I just read the notes of the EMA meeting and it looks like a green light for teduglutide. So that was really cool. And I was really fortunate because I was one of the experts that went to the FDA for the FDA advisory committee. And I'll never forget that day for many reasons. In fact, I rode to the NIH in a taxi with a woman who was on teduglutide who had short bowel syndrome, and she didn't know who I was. And she kept saying to me, if the FDA doesn't approve this drug, me and my friends are all moving to Europe because it's approved there. And the FDA, I was used to an FDA that was somewhat adversarial. The diabetes branch is pretty tough and they often push back and they're very focused on safety. And that's the FDA that I was used to. And that hearing, the company presented the data. The FDA advisors basically stood up and said, we agree with the assessment of the company surrounding the efficacy and safety of teduglutide. And I found myself sitting there and going like, are you kidding me? Like, that's all you've got to say, that you agree this drug works and has a favorable risk-benefit ratio? And that's how the day went. It was really amazing. I was on a plane to Italy that night to go give lectures in Italy, and it was a huge buzz. I got to say, it was amazing.
Nice bottle of Chianti on the other side.
I was getting emails telling me to upgrade myself to, a higher class of service, et cetera. But I was just so happy. It really didn't matter where I was.
So then let's transition to that because, in looking at your CV, you've got these 33 patents, and it's clear that you have been very successful at figuring out how to work at this intersection of basic science as well as translational medicine. So I'm sure that moments like that taxi ride were not the only times you've heard from really grateful patients. So has this aspect of your professional career, one that maybe you didn't envision from the beginning, been satisfying? What is it that keeps you in this line of work?
Yeah, so I'm always a clinician scientist when I wake up in the morning, and so the needs of patients and the unmet needs and the ability to make a clinical impact are always huge with me. So science is amazing, but being able to translate that into therapeutics is amazing to a next level. And seeing something go from discovery and patents and approval, I think, I don't know what the odds are, 1 in 10,000, 1 in 25,000. It's like winning a lottery. And so I always view myself as having won the lottery over and over again. And, there aren't that many people with short bowel syndrome that I meet in the supermarket or in the mall, but I certainly, met a lot of people taking the DP4 inhibitors, starting with my late mother-in-law who used to get terrible hypoglycemia on sulfonylureas and transitioned to DP4 inhibitors, and huge improvement in her life. And GLP-1, I think the last few years is really catching on. And in my own circle, family, people I meet at the golf club, friends, there are a lot of people now taking these GLP-1 drugs for type 2 diabetes and for weight loss. And it makes a huge difference in these people's lives. So I think more and more, one runs into people who are saying, I'm taking this medicine, and there's such an amazing satisfaction as being one of the, group of people that contributed to this.
I think I remember once when I was talking with Jeff Friedman, he had this quote of, for you, that this maybe wasn't so much lottery as the fact that luck favors the prepared mind.
Yeah, no, 100%, and there's variations on that. Like the harder I work, the luckier I get, or, better lucky than good, but really good to be lucky, et cetera. And I think there's no question that being at the right place at the right time, the DP4 inhibitor story. So I was giving a lecture at Tufts, Bill Bachovchin's in the audience, a world expert on DPP4, and boom, all of a sudden I have a collaboration. So that's not skill, that's just luck. And of course, one has to capitalize on and do the experiments and follow it through. And, the patents were battles. I can tell you stories about, we were sued by Merck, who were trying to overturn the patent portfolio because Januvia was doing so well and the revenues were really increasing that Merck was more inclined to bring more of those revenues in-house. And I was at the Prix Galien with Roger Perlmutter and Ken Fraser having drinks, and the thought came to me and saying, guys, what's with the lawsuits? Don't you guys have like enough success and enough financial sustainability, et cetera? But that's just part of the business. And so later on I sort of learned, if no one's suing you, you probably don't have a lot that's worthwhile out there. So it's just, initially it's really terrifying to get sued. After a while, it just becomes part of the business.
So I noticed from your CV, the more successful that scientists and physician scientists get, the more institutions heap committees and administrative details, and you've shouldered your fair share and taken on a lot of community service roles, editor-in-chief of different journals, and most recently organizing the Insulin at 100 conferences.
To be perfectly honest, I don't think I applied for any of these positions, whether it was endocrine division director or, director of the Banning and Betts Diabetes Center, editor-in-chief of Endocrine Reviews, running the Insulin 100 program. I can tell you that in all of those instances, I got called by a superior, a chair of medicine, the dean, et cetera, the president of the Endocrine Society. And in each one of those instances, I was sort of told or said, we think you would be a really great person to do this. And sometimes when you take these positions, it's very much a sense of, well, if I don't do this, who's going to do it? And will they really do an amazing job? Or should I just kind of, muscle up the energy and take this on? And there's also a sense, in the Jewish religion, we have this expression, tikkun olam, which broadly means social activism, repairing the world. But I think it's really important that all of us are members of a community, and we don't just want to be takers. We want to help build the community. We want to help provide opportunities. And it's hard to do that if you're just sitting in your lab hiding from the rest of the world. So none of the positions that I took were particularly onerous, to be honest. And, I just did what I could to make things move along when my turn came.
What do you see as the future for your lab for the next 5 to 10 years?
So, I have the most boring sort of research focus now. We're still trying to figure out how these glucagon-like peptides work. And, we have industry with phase 3 programs for NASH that a lot of people think are going to work based on phase 2 data. We have Novo Nordisk starting a phase 3 program for Alzheimer's disease and a huge amount of preclinical data for neurodegenerative disease, as well as clinical trials for Parkinson's with exenatide that suggest this might be promising. We have co-agonists that are in late-stage clinical development, amazing molecules like tirzepatide that might yet be the most powerful peptide therapy introduced for metabolic disorders. And to be honest, we don't really understand how these medicines work. So if I want to make the next great peptide therapy, I need to know, how do I dial in and preserve the cool stuff like cardiovascular protection, avoid any unanticipated adverse events, and make sure that I've got a really great effective molecule that's even better than what we have. And so I can just sort of see continuing to try and understand how these glucagon-like peptides work and help make them better in terms of the next generations. And that sounds pretty boring. There's not a lot of discovery science built in there, but it's going to have huge clinical impact.
If you could not have been a physician or a scientist, what other career do you think might have sustained you for these last 20, 30, 40 years?
So, I love almost everything that I think about or look at. So, I thought about being a lawyer for many years, maybe because I was told that I was argumentative and, would make a good lawyer. I like fixing things, although I'm not very good at it. I think accounting is cool. I've had tremendous exposure to the investment industry through interacting as a consultant to biotech or large pharma companies. I think working as an investment analyst is really cool. I love the financial services industry and creative financial instruments and investing and equity. So I'm not narrowing it down. There's just a lot of really cool things that one can do. I haven't mentioned professional athlete. I think Bruce Spiegelman had aspirations along those regards. I was not a very good athlete. I love sports, but I'm not very good at them. I'm participatory, but I don't excel. But I could have seen myself doing many different things. I love to read, and I think I have a tremendous family. My wife Cheryl is supportive. My kids are supportive. So who knows what I would have done, but I'm pretty sure I would have been happy and hopefully had some degree of success.
Well, thank you for that. I think we're all quite pleased that you did choose the path that you did. There's hundreds of millions of patients out there, certainly, who are very glad that you did choose your path. So thank you so much for joining me today, Dan. I really appreciated hearing a little bit more about the man behind all of those discoveries.
Thanks very much for inviting me. It was my great pleasure.